Welcome to Rapid mRNA Manufacturing for Vaccine Development. I am Jessica Madigan, Director of Business Development at TriLink Biotechnologies. Today, I will cover in my presentation a roadmap for planning and developing a successful messenger RNA program. I will discuss mRNA design considerations. I will provide an overview of the manufacturing process and the path to commercial scalability. And finally, I will cover important quality control assays for characterizing and releasing mRNA vaccines. These are our forward-looking statements. TriLink was founded in 1996, and since 2011, we have been pioneering and leading contract manufacturing of messenger RNAs. We were the first CMO in the world to offer mRNA synthesis services. In addition to messenger RNA, TriLink manufactures oligonucleotides via solid phase synthesis, nucleotide triphosphates, messenger RNA cap analogs, including our proprietary technology called CleanCap, and plasmids. We offer comprehensive CDMO and CMO services. We empower research, diagnostic, vaccine, and therapeutic programs with critical reagents, expert scientific consultation, and flexible support. Our manufacturing scales with program needs, from discovery to GMP grade. At TriLink, we do not have any of our own therapeutic or clinical aspirations. Whether you are an experienced mRNA user or perhaps using an alternative technology like inactivated viruses or viral vectors and would like to try mRNA, TriLink can provide a complete solution, many of which I will cover today. mRNA vaccines represent a promising alternative to conventional vaccine approaches because of their high potency, capacity for rapid development, and potential for low-cost manufacture and safe administration. Therapeutic considerations and challenges include optimization of your lead construct in the preclinical and toxicology phases, establishing manufacturing in a GMP environment for phase 1, and scaling up good manufacturing practices for commercial applications. For mRNA vaccines, TriLink supports our clients from development through phase 1 and eventual tech transfer for commercial. Vaccines elicit potent immunity against infectious diseases, as recently popularized by Pfizer and Moderna's SARS-CoV-2 vaccines. Here I am showing the common steps used in development of an infectious disease mRNA vaccine. In this example, I am highlighting a self-amplifying vaccine, but the steps are generally the same for traditional messenger RNAs as well. First, a sample is taken and the sequence is identified. Then mRNA, or self-amplifying RNA, is produced to express the antigen of interest. The mRNA is formulated and delivered to patients where it then expresses the protein, mimicking a viral infection and protecting against the virus. Other mRNA approaches to vaccines include immuno-oncology applications. Cancer vaccines have been employed in numerous clinical trials in which a tumor is sequenced in a patient and neoantigens that are only present in the tumor environment are identified. These neoantigens can be expressed in an mRNA to make a personalized vaccine. When delivered, the resulting expression of the mRNA recruits the immune system to target the tumor. Some trials have shown promising results of antigen-specific T-cell responses and prolonged disease-free survival in some cases. The remainder of my time today will focus on this one process, mRNA manufacturing. mRNA design considerations. So what would an optimal mRNA include? First, tailored activity and duration, whether the desired expression is prolonged or transient. Second, costs need to match the budget. Be sure to consider the long game here. Vaccines need to be low cost to be attainable for mass populations. The third bullet highlights where you can start to plan for financially attainable costs. You will need to consider the design of the DNA template for economic manufacturing processes. I will go into how to do this in later slides. Furthermore, you will need to consider various options on how to dial in an immune response with your product. I will touch on this later as well. Synthesis and purification processes need to be able to meet good manufacturing practice requirements. And finally, plan ahead for commercial. You will find that messenger RNA manufacturing processes that are suitable for small scale do not translate to large scale, and oftentimes have very different impurity profiles I will give you some options today on how we can address this concern. When approaching an mRNA synthesis, these are the key points of concern. Target selection. You choose your vaccine indication. Then you pick your protein of interest. This usually includes screening of multiple candidates. Unfortunately, I don't have a silver bullet for you there, but TriLink can make multiple mRNAs in parallel to fast-track R&D experiments. The 5' untranslated region, or UTR, is upstream from the coding sequence. Within the 5' UTR is a sequence that is recognized by the ribosome, which allows for the ribosome to bind and initiate translation. The 3' UTR is found immediately following the translation stop codon. The UTR plays a critical role in the translation termination as well as post-transcriptional modification. The N7-methylguanosine cap structure is usually required for ribosomal recruitment. Base modifications are important for eliciting immune responses. Poly-A tails are required for ribosomal recruitment. And the purification method needs to be defined based on the required specifications and ultimate scalability of the product. This phase often takes an average of 6 months to 1 year, but TriLink has solutions to expedite this process. As demonstrated in the previous slide, by leveraging advancements in NGS, we now have an effective, unbiased way to identify new disease strains and other pathogens without prior knowledge of organisms. Sequencing of entire genomes can take place in a matter of hours. From that data, you can select which proteins you would like to express, again, starting with the start codon and ending with the stop codon. Now you have your open reading frame. Selection of UTRs can be a challenge. There are many patents out there on how to do it. However, TriLink does have a streamlined and simplified option to expedite vaccine development. We have an entry vector that contains generic UTRs that have been proven to work well in messenger RNA therapeutics and have even been used in human clinical trials. For capping, TriLink's one-pot synthesis method with CleanCap is the superior capping choice as it yields a natural Cap1 structure at greater than 95% efficiency. Selection of modified bases is driven by the customer, but TriLink has one of the largest catalogs of modified NTPs to select from. Our entry vector has an encoded 80A poly-A tail, which has been shown to be very stable to recombination in E. coli DNA prep. And finally, TriLink has 3 platform purification methods available. By utilizing these tools, you can accelerate from 6 months to 6 weeks for a research-grade mRNA that can be used in ex vivo or animal studies. Plasmid manufacturing. To help our customers advance to the clinic quickly, especially those with little to no mRNA experience, we offer our standard entry vector that contains all required structural features for efficient transcription and translation of mRNA. Now, as I said before, we have created a simplified and streamlined process for UTR selection, but I would like to caution that we do not claim that all UTRs are equivalent. If your timeline allows for flexibility, I would encourage exploring various UTR sequences to optimize your product. Our entry vector contains the following elements: a T7 promoter that's compatible with CleanCap, a 5' UTR with a strong COSAC sequence, a 3' UTR derived from alpha globin, a poly-A tail with a variety of type 2S linearization sites to leave a perfect poly-A tail without any residual nucleotides. And finally, a kanamycin resistance backbone, as this is preferred over ampicillin. When manufacturing a plasmid for mRNA, you need to make sure the process is endotoxin-free, as mRNA purification methods are typically not endotoxin reducing. What you put in is what you will get out. You want to make sure that you are not encoding for various epitope tags. These can be very useful in an R&D setting, but for research, but are undesirable in a therapeutic. You will need to determine if GMP-grade plasmid is required for your product. Regulatory guidance suggests that GMP-grade raw materials should be used in the production of GMP-grade APIs and drug substances. Ultimately, it's the client's decision on what grade is suitable for their IND. On the same topic, master cell banking. Is this required? Again, from a regulatory perspective, master cell banking would be desirable for product control, but it is up to the client to decide. Plasmids can be a big bottleneck and should be considered as early as possible in your timeline. If you require master cell banking, industry timelines are usually greater than 6 months for establishment. In this table, I have provided estimated timelines for plasmid production at TriLink, starting from a cell bank. The shortest possible turnaround time is 3 weeks for GMP-linked plasmid, and the shortest possible timeline is 3 months for GMP plasmid. By choosing TriLink as a manufacturing partner, we will have the ability to accelerate your program timelines because we are a single source solution. There will be limited to no lag time between plasmid and mRNA manufacturing, condensing your critical path to the clinic. Our manufacturing scales start at 3-liter fermenters and go up to 30 liters. Here I am showing the standard quality control release strategy and specifications for our plasmid leading to mRNA manufacturing. It is important to test for process-related impurities, residual RNA, residual host cell proteins, and residual DNA, as well as percent isoform purity, endotoxins, and sequence verification. We acknowledge here that sequencing is the longest lead time. TriLink is currently leveraging outsourced partners for these services, but are looking to bring them in-house to further expedite release. Capping with CleanCap. At TriLink, we developed a novel co-transcriptional capping analog called CleanCap. It offers improvements over legacy capping technologies as it yields a Cap1 structure. Traditional capping analogs yield a Cap0 structure. Cap structures are important in self-non-self recognition. Cap1 structures are often found in higher eukaryotic messages. While Cap0 is sufficient for ribosomal recruitment, it can activate the immune system. Cap1 methylation reduces binding to pattern recognition receptors. IFITS recognizes non-methylated caps and sequesters them from the translational machinery. Additionally, Cap1 structures are more resistant to decapping enzymes. This results in the longer persistence of mRNA in vivo and greater protein translation. Another benefit of CleanCap is the increased yield from the manufacturing process. CleanCap yields more than 3 times that of ARCA or MCAP. And the one-pot transcription method will save time and money as compared to an enzymatic approach. CleanCap initiates transcription with a trimer nucleotide as shown here. The structure consists of an N7-methylguanosine followed by a triphosphate and 2 bases that are complementary to the first 2 bases of the mRNA transcript. The preferred embodiment of our CleanCap technology for a standard mRNA is adenosine followed by guanosine. ATP is not a good substrate for T7 polymerase to initiate transcription. And by having the first base of the transcript be adenosine, T7 polymerase will preferably incorporate CleanCap over ATP, driving high capping efficiencies without having to limit NTPs in the reaction. I would also like to share with you another version of the CleanCap technology called CleanCap AU that is specifically designed for self-amplifying RNAs. Self-amplifying RNA vaccine platforms are based on the genome of positive strand viruses, commonly called alpha viruses. These mRNA vaccines are based on an engineered viral genome containing the genes encoding for RNA replication machinery, whereas the structural protein sequences are replaced with the gene of interest, and the resulting genomes are referred to as replicons. These vaccines are capable of directing their self-replication through synthesis of the RNA-dependent RNA polymerase complex, generating multiple copies of the antigen-encoding mRNA, and express high levels of protein when they are introduced to the cytoplasm, in a way that mimics the production of antigens in vivo by viral pathogens, triggering both humoral and cellular immune responses. While there are no commercial saRNA vaccines, there are many in preclinical and clinical phases. CleanCap AU allows us to initiate transcription with an authentic AUG start of an alpha virus replicon. The capping efficiency is extremely high. A challenge represented by replicons is that they are extremely long, approximately 9 kb to 6 kb in length. T7 polymerase is highly processive, and the quality of the product from the transcription reaction is fairly high. But with each manufacturing step post-transcription, it becomes increasingly harder to maintain full-length product, as the molecule is extremely sensitive to physical shearing. The use of CleanCap AU enables one-pot transcription, one-step purification, which will maintain the integrity of the product. Enzymatic capping traditionally requires an additional difiltration step after transcription. Another issue to highlight, because of the highly structured nature of the 5' end of the replicon, it requires heating to disrupt the structure for enzymatic capping. Without heating, the capping will be inefficient, but this heat step will degrade the saRNA, making it a suboptimal choice for therapeutic manufacturing. We are very enthusiastic about CleanCap, and it has been very rapidly adopted. We can reduce the number of manufacturing steps from 4 to 2. We reduce the number of enzymes used in the manufacturing process, and the process is highly efficient, reproducible, and robust. Using CleanCap, we can accelerate the manufacturing process by multiple days, and the cost of manufacturing is lower than legacy cap analogs and enzymatic capping. Immune stimulation and double-stranded RNA. Several characteristics distinguish pathogenic foreign RNAs from self-RNAs. Location. Self-RNAs are in the nucleus or cytoplasm and are not found in an endosome. Transfection of cells with unmodified RNA can lead to cell death due to activation of innate immune pathways. Some cytosolic sensors include PKR, which senses double-stranded RNA, MDA5, long double-stranded RNA, RIG-I, 5' triphosphate, IFITs, 5' triphosphate and CAP0, CGAS, STING, cytosolic DNAs. Modifications of mRNA with pseudouridine reduce binding to innate immune sensors in vitro, reduce toxicity, and prolong expression in culture cells and in vivo, as demonstrated by Carrico and others. A major contaminant of in vitro transcription reaction is double-stranded RNA, an unwanted byproduct. There are 3 methods popularized to reduce the immune response caused by double-stranded RNA. The first is the use of modified bases, as popularized by Moderna and Pfizer SARS-CoV-2 vaccines. The second is through purification to deplete double-stranded RNA post-transcriptionally. The third is to optimize transcription reaction to avoid the generation of the double-stranded RNA in the first place. For the first option of using modified bases, it's important to note that the use of certain modified bases are covered by intellectual property and licenses may be required. In order to avoid IP concerns, you can use reverse phase HPLC purification. This process is a burdensome procedure requiring special instrumentation and generating hazardous waste. It also requires high temperature to disrupt the secondary structure for efficient depletion of double-stranded RNA. This high heat can damage the mRNA product and it limits its scalability. The third option is likely the best path to success. Each RNA sequence is unique and may require unique conditions to achieve low double-stranded RNA content. And it requires time and money at the initiation of the program to complete optimization, but it can pay dividends in the long run. TriLink has experience and knowledge on the best transcriptional conditions to drive highest yield, purity, and capping with low double-stranded RNA content. On the right, you can see an example of a slot plot with 9 different transcription conditions in triplicate to drive low double-stranded RNA content. mRNA manufacturing. We can customize the mRNA manufacturing process to meet each client's needs. In general, the process starts with a circular plasmid that is digested, then purified. This is followed by in vitro transcription with T7 polymerase, pyrophosphatase, RNase inhibitor, NTPs, and a cap analog. The RNA is then treated with DNase to degrade the DNA template. We then continue to any post-transcriptional enzymatic treatments like capping, tailing, or phosphatase treatment. Then purification and 0.2 micron filtering. Purification of mRNA is required to remove salts, NTPs, cap analogs, proteins, DNA, and possibly truncated mRNA products and double-stranded RNA. Options include lithium chloride precipitation, which results in a crude product which is not scalable, silica membrane spin column purification, which results in a pure RNA product but is also not scalable. LC isolation results in a pure RNA product and is scalable to grams and possibly beyond. Oligo-DT results in a pure full-length RNA product and is highly scalable. And reverse phase HPLC results in depletion of double-stranded RNA contamination, but is limited in scalability. Commercial biomolecule production is going to rely on process chromatography. Here's more information on the 3 chromatographic methods of mRNA purification that TriLink has developed. LC isolation is a cation exchange method that enriches for pure RNA. We consider it a scaled-up equivalent of silica membrane as it removes proteins, DNA, and other transcription residues. It does not separate RNA by size, and it does not remove double-stranded RNA. Reverse phase HPLC will fractionate by size and is used to deplete double-stranded RNA contamination, but as noted before, it uses high heat, high pressure, and volatile solvents. It presents difficulties with scale-up, and it can only be used on sequences that are shorter than 6,000 bases. Oligo-DT is a highly scalable and recently popularized method for mRNA purification. It captures the RNA by the poly-A tail and results in higher purities of full-length product. The average GMP manufacturing process takes 5 to 7 days. Purification can be the biggest time suck, especially if the RNA is purified by reverse phase HPLC. This is where it has been the most important to create optimized and scalable processes for commercial manufacturing. At TriLink, we are focused on developing a platform, large-scale manufacturing process that is optimized for commercial scalability. Legacy manufacturing processes used water baths, which have poor mixing control and limited volumes of less than 500 mils. We have invested in bioreactors that can be used for enzymatic reactions. We have developed scalable solvent-free purification processes. We have optimized tangential flow filtration loading capacities and optimized the sizes of molecular weight cutoff filters based on the RNA lengths. We have also expanded our purification processes to include the oligo-DT purification resin. You can accelerate to the clinic by taking advantage of our platform manufacturing processes. However, it's important to note that difficult secondary structures and various lengths may require further optimization. The bioreactors that we use at TriLink are commercially available instruments that scale from 100 milliliters to 500 liters for easy tech transfer. They have a high degree of mixing with low shear, and at TriLink we offer 100 mils, 500 mils, and 3-liter options. A quick summary of the benefits of our optimized platform manufacturing process are shortened manufacturing timelines, higher purity product, exclusion of solvents, one-pot synthesis, one-pot purification, and an easy path to tech transfer and scalability for commercial manufacturing. mRNA quality control. At TriLink, we offer a large panel of assays to characterize messenger RNA and process-related impurities. On the left side of the screen, you can see a number of non-compendial assays developed for final release and in-process controls of mRNA vaccines. On the right are general compendial methods that are common for API and drug substance release. For the last part of my presentation today, I will go into more detail on the mRNA-specific assays. Now, in order to assess the capping status of an mRNA, which is actually quite challenging because we are looking for the addition of a single methyl group to an RNA that can be millions of Daltons in length, we developed a capping assay at TriLink in which we cleave the 5' end of the transcript and then use LC-MS to detect for the heterogeneity of the 5' end. On the right-hand side of the screen, I have 2 examples of capping assays. One is an enzymatically capped product that has not gone to completion, and the other is a clean cap transcript. In order to assess the poly-A tail length, we have developed another LC-MS method. First, we digest the RNA product, leaving the poly-A tail intact, and then run an LC-MS. As you can see here, we take the deconvoluted spectrum and relate it to the intensity to determine the poly distribution. Residual protein assay measures the amount of residual protein present in an mRNA sample after purification. Residual protein is typically measured by nano-orange or silver stain. Silver stain is highly sensitive but qualitative in nature. We normally would use silver stain to optimize protein clearance in process and recommend using a qualified and quantitative method of nano-orange for final release. A residual plasmid DNA assay measures the amount of residual plasmid DNA present in mRNA samples after purification. This method uses a dual-labeled probe and real-time PCR reaction to measure the amount of amplicon present in an mRNA sample by comparison with a standard curve generated from known amounts of input plasmid. We have platform assays developed for detection of residual DNA against a kanamycin resistance backbone, ampicillin resistance backbone, and TriLink's 3' UTRs. Custom primer and probe sets can be designed as well. For assessing the purity of an mRNA, we offer a variety of different methods, including bioanalyzer, agarose gel, fragment analyzer, and ion pairing reverse phase HPLC. Fragment analyzer is the most common method used for final release. A fragment analyzer is used to establish RNA integrity by using the Agilent Fragment Analyzer instrument. This system utilizes automated parallel capillary electrophoresis to provide quality control for nucleic acids. Purity is established by smear analysis as compared to a reference RNA. Ion pairing reverse phase HPLC was derived from its parent technique, reverse phase chromatography. It uses hydrophobic stationary phase and a mobile phase with varying hydrophobicity depending on the ratio of polar and organic solvent. It is an effective method for evaluating the integrity of mRNA. In this slide, I am showing data of a firefly luciferase mRNA that has been purified by oligo-DT. For the fragment analyzer, the blue trace is the material after first difiltration but before purification. The red trace is after oligo-DT purification, and the black trace is final product. For IPRP HPLC, the blue trace is the material after first difiltration but before purification. The black trace is after oligo-DT purification, and the pink trace is final product. Developing specifications for the manufacture of an mRNA vaccine requires a comprehensive and critical analysis of the requirements that are used in developing and validating sound analytical methods. The approach to setting specifications for mRNA is not straightforward, as the molecules are complex. Because of this, it's important for CMOs and the sponsors to have a process-driven approach to specification development in order to meet both FDA and ICH standards. At each stage of development, from phase 1 through securing FDA approval, companies need to ask themselves what is absolutely necessary to ensure the safety and efficacy of the drug. Ultimately, the customer will define what specifications must be met to maximize likelihood of regulatory acceptance. It's also important to note that amounts of material required for final release and retention samples may actually exceed the amount needed for a study. So you need to start thinking early in your program about what release tests need to be run in order to plan ahead for the eventual manufacturing scales needed for production. TriLink has committed to meeting and growing with the needs of our customers and the mRNA vaccine industry. We have a 118,000 square foot facility in San Diego, California with expansion opportunities. And in the last year alone, we have added 10 additional ISO-classified clean rooms to support increasing demand and added plasmid manufacturing services. Thank you for your attention today. I hope I have provided you with sufficient information on the important considerations for mRNA vaccine manufacturing and the ways you can accelerate your vaccine development with TriLink. Please do not hesitate to contact us with any follow-up questions. Thank you.